My dear students, welcome. It is my pleasure to invite you to the massive open online course on Principles of Biotechnology. This is an online course for 2 credits covering 22 modules where I am sure that we can have an interactive session as well included in the program. The course is based on choice-based credit system of University Grants Commission. This course on Principles of Biotechnology is offered to you by the Educational Multimedia Research Center of Madurai Kamaraj University. This course is intended to the undergraduate students of any life science discipline and those individuals who are all interested in knowing the fundamentals of biotechnology can also take up this course, which will help them in doing their specialization in biotechnology in the near future. Well, dear students, friends, what do you think of when you hear the word biotechnology? Well, maybe the things you have seen and hear from the news, such as the genetically modified organisms like the cloned sheep Dolly, Polly the goat, and Andy the monkey, the transgenic plants like BT cotton, BT brinjal, and BT bendi, the human insulin, humulin from Escherichia coli, and as well the human genome sequencing project, the plant Arabidopsis thaliana and rice genome sequencing projects, and the insect Drosophila melanogaster genome sequencing project. If that is what you think of, yes, you are absolutely right. Of course, these are the products of biotechnology. But what about beer brewing, hybrid plants in crop breeding, and antibiotics penicillin? These processes and products, some of which have been around for thousands of years, are also examples of biotechnology. And actually, biotechnology is an interdisciplinary, recent, and an young discipline of science comprising the principles, methods, and approaches of the classical disciplines of genetics, plant breeding, biochemistry, microbiology, and the modern disciplines of molecular biology and information technology. Of course, the latest area of genomics, proteomics, and other omics too. In this particular course on Principles of Biotechnology, we are going to familiarize with the basic knowledge of tissue culture, molecular biology, recombinant DNA technology, plant transformation, and immunotechnology in 5 sub-themes. Many an occasion, new discoveries in biology have refashioned the trends of thoughts and comprehension. Such landmarks contributed by several brilliant minds need to be known, particularly in the context of the beginning and growth of biotechnology. And so this course starts with the chapter of revisiting the history and concepts of biotechnology. Well, the success in understanding of biotechnology principles lies with the awareness of the in vitro culturing of plants through tissue culture principles and techniques. Hence, the first sub-theme of this course makes you to understand the nutritional requirements of tissue culture, the concepts of morphogenesis, and the basic types of tissue culture techniques. The second sub-theme focuses on the knowledge of structure and function of DNA. So the second sub-theme, basics of molecular biology, was designed in such a way to give you an overall understanding of the nucleic acid structure, DNA processes, the basics of a gene, and the DNA sequencing methods. At this point, I can say that biotechnology involves the principles, practices, and methods of molecular biology and tissue culture. And the recombinant DNA technology is the tool to combine the set 3 to create a hybrid DNA of the gene of interest and the vehicle DNA, which leads to the transgenic plant production. Hence, the third sub-theme is rDNA or recombinant DNA technology, and in which we are going to explore the fundamentals of recombinant DNA, examining the vector elements of DNA, manipulating DNA enzymes, and the construction of recombinant DNA molecules. Fourth sub-theme is the plant transformation, and it has rooted through the natural process of bacterial transformation. Production of transgenic plants depends on the plant transformation methods and successful selection of the recombinants amidst the non-transgenics which are to be eliminated. And it dealt with designing plant transformation, familiarizing with reporter genes and promoters, analyzing methods of direct gene transfer and indirect gene transfer. Microbiological principles and production of antigen and antibodies are the basic needs for the selection of transgenics in any transformation procedure. And the modern hybridoma technology is the latest achievement in microbial recombinant technology. Hence, the fifth sub-theme is the immunotechnology, in which you are going to be introduced to the structure of antibodies, identifying the structure of an antigen, pairing antibody and antigen, familiarizing with monoclonal and the polyclonal antibodies, and applying hybridoma technology. And the outset, I have no doubt that this course content is very well prepared to suit with the purpose of familiarizing biotechnology principles to the non-familiar students of various disciplines of life sciences. The course is designed in such a way that you can have the access to go through ebook, objectives, summary, glossary, case study, tutorial, FAQs, transcription of video lectures, and assessment. Being a student, you can discuss with the subject experts for further clarification. To test your understanding, there will be a final assessment paper in the form of question bank, which includes subjective or objective questions at the end of the program. After the successful completion of the course and taking up the assessment, the certificates will be provided by the host institutions for the eligible candidates. Hope to meet you all aspirants of principles of biotechnology soon. All the best.